you. Thank you, everyone. Today is my great honor to share our research on how we develop a machine learning model to predict ADME properties of chemicals and nanoparticles. In my presentation, I will start with an overview of the application of machine learning model in the field of toxicological sciences, especially in PBPK modeling. And then I will show two recent publications as example of application. And then I will discuss the current progress and the challenge in this field. Our lab is a computational dry lab that we develop computational models to study food safety, chemical and nanoparticle risk assessment. We can develop different types of models, mainly focus on PBPK and QSAR models. We can develop models in a number of different animal species and in humans at different levels. In recent years, to build more robust model, we incorporate machine learning and AI algorithm into our models. PBPK model is a computational simulation process that describes the absorption, distribution, metabolism, and the excretion of chemicals, nanoparticles, and they are metabolized in the body based on the interrelationship among the key physiological, biochemical, and physicochemical properties using mathematical equation. PBPK models are very important from the perspective of risk assessment because they are the model that can predict the target organ exposure based on environmental or occupational exposures. In the field of toxicology, one fundamental principle is that both the beneficial and the adverse response to compounds are related to the concentration at the target organ rather than the concentration at the site of absorption. So in the field of toxicology and risk assessment, we really need to have some tool such as the PBPK model to predict internal target organ exposure. QSAR modeling is a modeling method to study the relationship between chemical structure and the biological properties of chemicals. In our lab, we mainly focus on building QSAR model to predict absorption, distribution, metabolism, and excretion. Both PBPK models and the QSAR models have been commonly used in toxicology, risk assessment, drug discovery, and development. So both are important tools in future chemical risk assessment. There's no need to define AI because it has been defined in many excellent previous public presentations in this workshop. We started to incorporate machine learning method into our model a few years ago. The first step is to conduct a comprehensive literature review. We found that machine learning and AI method being applied in different subject areas of toxicology, including to predict toxicity, to incorporate, to integrate with toxicogenomic data, to identify potential mechanism of toxicity, and also to predict ADME property, even to develop a PBPK model. And we found that for the ADME and the PBPK domain, there are so many studies that are sufficient for a separate review. So we write another review that particularly focus on PBPK modeling. In our review, we analyze existing methods that have been applied in this field. Throughout today's presentation, most of the studies have been using deep neural network, which is one type of machine learning method. In the literature, there are many other studies that have applied other more traditional, simpler machine learning methods. We have summarized the limitation and the advantage of each of these methods. And these methods are listed here. And then we also found many studies have been published to predict different types of toxicity endpoints, including in vitro toxicity and in vivo toxicity on different organ systems. And this table just lists several representative publications. And this table is for pharmaceutical compounds. We also summarize many studies on machine learning model to predict absorption and distribution, as well as metabolic and elimination properties. This could include human intestinal absorption rate constant, partition coefficient, and like maximum metabolic rate, and then renal or total clearance. This table is for pharmaceutical compounds. In our review, we found that there are more publications related to pharmaceutical compounds, but there are relatively limited studies focused on non-pharmaceutical compounds, which are mainly environmental toxicants in the domain of ADME prediction. And one prerequisite to build machine learning model is to have a good quality data set. So in our review, we also identify a list of databases that contain PK data that are suitable for machine learning analysis. And this table only lists some representative database focused on PK. And this table lists representative database that contain different types of toxicity data that can help future machine learning model development. In our review, we see the great potential of integrating AI approach with traditional computational models to develop a more robust model, and the machine learning and AI approach has substantially driven the toxicology field to move forward. However, we also identify some data gap. One of the data gap is there are many models to predict ADME or toxicity property in rodents and humans, but very few studies focus on food-producing animals, which are also relevant to human health, especially food safety assessment. And another data gap, we found that most of existing AI-based model, mostly for small molecular drugs, relatively fewer study on environmental chemicals, and is even very limited for nanoparticles. And this data gap guide our recent studies. So next, I will move on to the first example. Is we develop a machine learning to predict ADME properties of chemical. This model is focused on food-producing animals. In the field of veterinary medicine, animal will require a withdrawal interval when they receive a drug at a labeled dose. However, in this field, because there are not so many drugs for veterinarians to choose, many times they have to give a drug in an actual label manner, which means to give a drug at a dose that is different from the label. After giving an actual label drug use, they need to prescribe an actual label withdrawal interval. The challenge in this field is we don't know the actual label withdrawal interval after actual label use, because the actual label use could be so different dose, different frequency, different number of injections in different food animal species. In the US, the US Department of Agriculture, they have been supporting a program called the Food Animal Residue Avoidance Data Bank, with the objective to help producer veterinarians to prevent or mitigate illegal or harmful residue of drugs, environmental chemicals, or other chemical agents that may contaminate the foods of animal origin, with the ultimate goal to provide food safety, protect food safety. Currently, there are 5 regional centers, and I have been working in this program for more than 10 years. I was the PI in the Midwest Regional Center at Kansas State University, and now I'm the co-PI at the University of Florida. Different regional centers have different responsibilities, but we work together collaboratively. In my lab, my major responsibility is to develop PBPK and QSAR models to help predict the PK properties of different drugs in different food animal species, so that the responders can use our model to predict the actual label withdrawal time to help answer requests from veterinarians, ultimately to help protect food safety. We have been working in this field for more than 10 years. In the first couple of years, we developed simple PBPK models for drugs in cattle and swine. But at that time, the model was simple. It didn't consider population variability. Later on, we incorporate population variability so that the model can be used to simulate the kinetic for a diverse population of animals, so that the model can be used to predict actual label drug use and the actual label withdrawal interval. But at that time, the model was some complex computer code and not user-friendly. So in recent years, we convert our model to graphical user interface that are now easily used by responders and regulators, risk assessors. Also, in recent years, we incorporate machine learning model to build more robust model because we receive thousands of visual interval requests every year, and these requests are on different drugs, different environmental chemicals, different food-producing animal species. By developing one model for one drug is no longer fast enough to meet this food safety need. So in coupled machine learning model, our long-term goal is to build AI-assisted PBPK model to predict PK property of drugs and the environmental chemicals in food-producing animals. The framework of this study is we leverage the data from the Federal database, which, as far as I know, is the largest database on the PK of drugs in animals, especially in food animals. So we can get PK data such as time versus concentration data, half-life, clearance from this database, and then we extract the structural data from chemoinformatics software, and then we use a different machine learning model to build a good model to predict the PK property. As a starting point, our first project was to build a model to predict the plasma half-life of drugs after intravenous injection. In our database, we have more than 11,000 plasma half-life data points from the Federal database. After filtering all the data through different criteria, we narrowed down our database to only 341 data points for the plasma half-life in 6 selected major food animal species. And then we use this data to, we split the data to training set and test set, and then we develop a model to predict the plasma half-life of drug in this selected food animal species. We tried different types of molecular descriptor, which are listed at the bottom of the table. And we also tried different machine learning methods like the k-nearest neighbor, random forest, support vector machine, and deep neural network. And then we compare the performance. We found that the deep neural network model has the best performance with the most accurate prediction of plasma half-life of selected drug in these food animal species. To make our model more user-friendly, we convert it into a web dashboard. With this web dashboard, other researchers, they simply need to select a drug or enter the case number of a drug, and then they can quickly predict the half-life of the drug in the selected food animal species. And this model is really helpful for the Federal program because now if we receive a request on an unknown drug or chemical, we can use it to quickly predict a tentative half-life to help predict X-label withdrawal interval. And I also put this study from US EPA. It is also a study that tried to predict the plasma half-life, but this study was based on a limited data set because of the limited data available. And the good thing of this study is they were able to build a model based on only several dozens of data points as a categorical model. They divide the plasma half-life into several range or several beings, like less than 12 hours, less than 1 week, less than 2 months, and then longer than 2 months. And then they were able to predict the possible half-life range of a new PFAS. And so by comparing to these two studies, if we have sufficient data, we can build a regression model that can predict the exact half-life value. But if we have limited data, at least we can predict the half-life range of the chemical. So this is the first application. The next application is to show how we use machine learning model to build an entire PBPK model that not only can predict the plasma half-life, but to predict the entire kinetic profile. And in this example, we focus on nanoparticles. Nanoparticles have many biomedical applications. They can also be used in consumer products. However, in that field, there was some challenge. One is lack of robust computational tools to assess the risk of nanoparticles. And in terms of nanomedicine, one challenge in this field is a low delivery efficiency of the nanomedicine to the target site tumor. So our research is to build model that can hopefully help address these two challenges in this field. We started this project about 10 years ago. The first thing is, again, to do a comprehensive review on the PK property of nanoparticles. We found that the PK property of nanoparticles are quite different compared to small molecules, because when we build PBPK models for nanoparticles, we have to consider many other factors like the biocolonial formation, cell-specific endocytosis, dissolution, aggregation, etc. So it's more complex. At that time, we tried many different methods, and we finally found a multiplier Hugh function can stimulate endocytosis of gold nanoparticle. And by using this method, we were able to build the first PBPK model for gold nanoparticle in mice. And then later on, we extrapolate into rats and pigs. And then we extrapolate the model from each animal species to humans to support human health risk assessment of gold nanoparticles. And then to help address the challenge in the field of nanomedicine, we extrapolate our model from healthy mice to tumor-bearing mice by adding a tumor compartment. And to rigorously evaluate and validate this model, we did a very comprehensive literature search. We collect, in our phase 1 literature search, we collect more than 300 data sets from 200 publications, and then use this data to validate our model. And then after we have this model, it can simulate a kinetic profile of about more than 200 nanoparticles for different dosing regimens. We use the model to generate a lot of simulated data, and then we build machine learning model to predict the delivery efficiency based on the physical-chemical property of the nanoparticles. This is a simple data-driven approach. And then we also apply a hybrid approach, which is a combination of a data-driven approach and a physiologically based approach. In this study, we use our nanotumor database. We use this data to build a QSAR model that can be used to predict the critical PBPK parameters related to the tumor compartment. These parameters include the maximum update rate, release rate, and the Hugh coefficient. And then we incorporate this parameter into a PBPK model framework so that we can use this, we call it as AI-assisted PBPK model to predict the time course profile of nanoparticle in a tumor site. And this slide to show the correlation of our AI model predicted PBPK parameter related with the original parameter. We see pretty good correlation with a coefficient of determination of generally more than 0.7. And this is to show our model predicted delivery efficiency at 24 hours, 168 hours, and the maximum delivery efficiency, their correlation with the observed data. We see very good correlation at 24 hours and the maximum delivery efficiency, but the delivery efficiency at 168 hours, which is 7 days after injection, was no good. This is mainly because our training data set are limited to the early kinetic profile, which usually measure at 24 hours after injection, but very few data sets that provide data up to 7 weeks after injection. So this is a limitation of the model, but also identify a data gap in the literature. And this is a slide to show some representative result that our model can predict the entire kinetic profile of different type of nanoparticle in a tumor, and that correlated with experimental data quite well. So for the nanoparticle study that I present so far, the previous study all focused on one compartment, which is the tumor. However, from the risk assessment perspective, we really need to model that can predict the target organ dosimetry, especially at different organs like liver, kidney, and the spleen and the lung, where nanoparticle may accumulate in this organ because they contain a lot of macrophage where they update the nanoparticle into these organs. So to address this issue, we conduct another literature search to collect more data, not only the tumor data, but also data in the major organ, including liver, spleen, lungs, heart, and kidney. And we have collect a total of more than 10,000 data points to train our next model. So with this updated database, we develop another machine learning model in which we can predict the delivery efficiency to both the tumor and the major organs, including kidney, liver, lung, and based on physicochemical properties of the nanoparticle. And this slide shows the correlation between the prediction and the observed data for both the training set and the test set. So we also can do prediction using different machine learning algorithms, and then we again found a deep neural network model had a better performance than other traditional machine learning model. And then we also do feature selection to analyze which of the physicochemical property play a more important role in the distribution to tumor and to other organs. We found that the material type, MAT in this figure, has a consistent result that play a high important role in the distribution to both tumor and the major organs. And this figure also shows that these different factors play differential roles in the organ distribution. So when we design a nanoparticle, we need to consider all these factors in a model. Lastly, we convert our model to a web dashboard again. So with this web dashboard, the nanoscientists, they just need to input some basic physicochemical property and experimental study design factor into this web dashboard, and then they can quickly predict the delivery efficiency to tumor and the different organs. This will hopefully serve as an alternative method to help preclinical study of nanomedicine, hopefully to partially replace and reduce some animal study in the field of nanomedicine. And this modeling method can be applied to develop a model in healthy animals, which is important to provide a tool, PBPK model to help risk assessment of nanoparticles. So through our research, we found that nowadays we can leverage machine learning method to build many advanced QSAR models to predict ADME properties of hundreds, thousands of chemicals. We can also build AI-assisted PBPK model for hundreds or even thousands of both chemicals and nanoparticles. And then we can also analyze a large amount of different types of data to generate new insights into the toxicity mechanism rapidly, which was difficult by manual approach in the past. However, we also have found some challenge and a scientific gap in this field. So to build a validated machine learning model, one prerequisite is to have good quality data. So rigorous data curation, quality check, infrastructure to store, analyze, share data is important. Also, many of existing studies, we found that they usually try different machine learning methods to determine the optimal model, because different studies have different type of data. It's not sure which specific machine learning model may be best for a particular type of data. So we advise to try different machine learning methods to determine the optimal model. And then most of the existing studies, they are classification model to predict toxicity with a yes or no answer. In the future, hopefully we can see more and more models that can predict the dose-response relationship or the intensity of effect. And then we encourage other colleagues to convert their model to user-friendly interface so that more users can use this model. And existing machine study in the field of toxicology are mostly on rodents and humans with very few info animals. So this is a field that is underdeveloped. Hopefully in the future there will be more study in this field. And then there are many studies for small molecule drugs, like earlier presentation are mostly about small molecular drugs and chemicals. And but right now very limited study for ADME prediction of nanoparticles, which is another data gap. In addition, now we all see the strength of AI method in toxicology. I would say it's important for education and the training program for the future toxicology students. Like in our department, we have a course on AI in machine learning and AI in toxicology and environmental health, with the hope that future students know the AI principle and know how to apply it. And then we also writing and editing a book on machine learning and AI in toxicology and environmental health, which hopefully becomes a book for future generation of toxicology students. Last but not least, I would like to acknowledge my current lab member, former lab member, my collaborator, my advisor, and the founding agency. A lot of work I present today was done by my lab members. Thank you.